right, so today we're going to finish up the Pico lab. We're also going to go into a little bit more of the DNA stuff. We're going to talk about TAC if we have time. If not, we'll do that next time. And then we're going to do an activity. So I have not given you notes for this, so you're going to take your own on these. So grab out a piece of paper, and we're going to talk about some DNA sequencing. Information contained in the DNA sequence. I am recording it, so if you want to look at that later, you can. I will see about putting up a PDF of it also for those that want to just look at the PDF and don't want to listen to my voice. Okay? So if you grab out a piece of paper, we'll get started. We're going to play real college where I don't give you no cheat notes.
Before you start today, can I just say something? Before you start today, can I just say something? Stop.
I was waiting for you to say something, Goose.
I mean, I can.
Huh?
Instead
All right, here we go. Everybody got paper? Anybody need paper? You have extra paper up here if you need it. It's old, ancient, but it works. Okay. So we're going to talk about DNA replication first of all. What we've done is we've actually made plasma that we've inputted the globe to, and hopefully when we go back there we'll be able to see that. And we're going to take a little piece of it and put it into some broth, and so we can grow it, hopefully a bigger amount of it. Okay, a lot of you, okay, so we do a reagent mix for DNA replication. We have normal N bases and dideoxy N bases. And the dideoxy N bases are missing an oxygen for bonding for the next nucleotide. That terminates the chain. Okay? And it's radioactively tagged. We have within this reagent mix also DNA polymerase, a primer, and some buffers and salts. So we're going to be using this to make other things coming up. Okay? When a DNA strand termination in a dideoxy nucleotide cannot be elongated because it's a 3 to an OH is necessary for polymerization. And polymerization just means making it more and more and more longer and longer and longer. Okay? When we look at DNA sequencing, the Sanger method is what we're going to be using. It determines the nucleotide sequence of the DNA. We synthesize complementary strands of DNA in vitro using tagged bases. So as we look at a tagged base, this would be a tagged base, we can put in different things. We're going to be preparing different tags to be able to do different things. We have what they call ladders, where we know how long the fragments are, and they'll be part of our ladder that we can pair to other things that are next to it. Okay? Dyed nips will stop DNA elongation. When a dyed nip is incorporated into a new DNA strand, it stops it from growing any bigger. This is because D-nips lack the 3-hydroxyl and hydroxyl group, which is necessary to form the phosphodiester. So we don't get those diester linkages that keep adjoining nucleotides together.
Why is it pronounced dynips when it looks like bib-nips?
Bib-nips, dynips. How do you want to say it? Okay, so when we're reading the sequence, this is actually a sequence of a DNA that they did before. Yeah, only this is a lot bigger, longer gel. So they had the wells up here, you can see the wells, and this is when it actually has run that far. Okay, so we love the gel. You guys have done a gel before. And now we're going to be able to determine an A, a G, a T, a C, okay, the genetic code. And we can be able to read lanes manually. And you sort of see how it's reading right here. So G, A, C, G, C, T, G, C, G, A. So we get to this long chain right here, whoops, that we're able to keep going, keep making the chain. Okay, Fred Sanger, this was his second Nobel Prize. His first was in 1958 for the protein structure of insulin, and they actually gave that patent to the public, and so there is no patent on insulin. The money that it's costing right now for people to get insulin is because of the, not because of the insulin, but how it's delivered. So the pens and stuff that they're using to deliver it. The insulin itself is not expensive to make. Okay, here's how they've advanced it. Now they actually have a computer that can read it, automatic reading, so they didn't have to look at all those bases by themselves. They have a computer reading. And you can sort of see in the greens and the different colors what ones come when. And by how they come tells us what the sequence is. And so this is more automated. Each base has a different color.
How is it reading it? Like,
So it's still doing, it's still sort of like a gel kind of thing going on, but they're just reading, is it in this lane or this lane, by how fast they're going. Okay? And we'll be doing some of that. We've got some advances to sequencing. They did, they don't pour gels anymore. Okay? They've got a higher and faster kind of thing that's sort of a, it's sort of like a filter like thing like this. I don't know how to describe it, but it has 384 lanes, so it can go really, really fast. Okay, big labs are getting to do it faster and cheaper. We've already talked about the Joint Genome Project. There's a Joint Genome Institute. MIT is working on it, Washington University, Baylor College of Medicine, Sanger Center in the UK, and Celera Genomics. This is a big kind of scale, so you've got to think big scale. So these are like those little tubes. They're capillary tubes. They're very, very small, and along there, it's forcing it through, and as it forces it through, they have a detection window that tells what is coming through. Is it a longer strand or a shorter strand, or what's coming through? So it starts all here together, and then as they separate, we get the reading. Okay? So 4 reactions of primer, DNA, polymerase, we get the A, G, C, and T. With radioactive dye, without a 3 oxygen end, it can't make it longer. And then we do the electrophoresis of the fragments. With the electrophoresis of the fragments, we end up getting different things that are able to be sequenced. And they're sequenced according to how long their fragment is. So on that electrophoresis, the longer fragments take longer to make it from one end to the other. Shorter fragments, not so much. So as they're broken up, we can see where, you can see how this is still the same thing, it's just got a couple more things added to it. So we're able to see what the next step is. So here we can say, okay, this is the longest fragment, and go from there. Pretty easy. This is a long gel that they used to have to read just by hand.
Nice.
This is where they get it automated. Now that they've got it automated, they say, oh, if it's this color, then it's red. Or if it's red, it's T. If it's whatever. And they're able to have the computer, it's down here below, tell what it is. Okay. So, that's it.
You go back.
Yeah. Maybe. Maybe not. Didn't want to come back.
It's okay. I know.
Got it?
Yeah.
So that was the last one, I believe, right there. Okay, so what we're going to do is we're going to go back. We're going to check our,
Do you have a black light?
I do have black light. We're gonna check and see if we got them to glow. If we got them to glow, it's good. We'll go to the next part, which is you'll need to get an inoculation lube. You'll need to get one of those conical tubes and fill it. You gotta listen. Okay, we're gonna get a conical tube. We're gonna fill it with some nutrient broth. It has to be the nutrient broth with the agros. Okay? And you're going to take one of the colonies and you're going to put it into the broth. Shake it up. We're going to put it into a test tube rack, and we're going to put it on the shaker. And it's going to shake until it gets mixed up. And hopefully it will grow while we do that.
Like 2 days.
Yep.
That's a lot of days.
Yep, so we'll see what we get. Okay, so, and then we've got an activity that I printed off at the back. So let's go ahead and get started back here and see how we do. Gloves.
On the, it was like the first 2 slides, it was where it's like tell you about the end bases. Will you go back to that one?
If you like it so much.
Which one would you like?
That one.
This one?
Yeah, that one. Okay, what?
Okay, show me. Awesome.
Yo, look at that. Look at that.
I've
I'll see you.
I've just been
All right, who's the flex this
today,
time?
and I was like,
Unbelievable, huh?
what's going on? Just kidding. I just talked about it.
How's the grades for you? Pretty good.
Everything.
Oh,
The
really?
DJ was so much better for
Really?
starts than
I
Harvest.
suck.
So
got-
I have a
Literally, our
question.
DJ sucks. I'm
We
sorry,
got
guys.
a new
I don't
DJ.
know.
Which
So I have
is
a
I
question.
better. I didn't go to Harvest.
Yeah, no, Harvest sucked.
It was like Harvest.
Did you
Oh
feel
no, Harvest
I just what got me
Yeah.
was when JC threw up.
Harvest
Yeah.
played
And
like
then she just
Sweet
I
Caroline.
didn't go back.
didn't hear about it till after.
You
I
played
didn't
that. You
go
played.
back.
They played like
She was like,
well-known
"Just
songs,
keep
but
doing
not
it."
dance songs. Like they played Sweet Caroline, and you can't like dance to
You're
that.
so
I don't know. I
Mosh
just like
pit,
I didn't
you know.
I didn't go back into the mosh pit.
And then he would like every like 30 minutes, he was like,
And she was
Alright,
like, bouncing
look
off
at
the
that,
wall.
how
I was
we
like,
doing?
girl, you gotta sit down.
we're like, whoa.
Yeah.
I know, this was talked a lot too.
Guys, and that was
They
it.
didn't stop. I would have been embarrassed.
Yeah.
Did you just turn the thing up?
After I got there, he would talk.
Determine the nucleus.
Yeah.
Stop talking.
I would have been embarrassed.
You don't need to
talk.
No,
And then she like
It was so much
just
better.
everything
I know,
saying
I
it.
was
I was like,
I mean, never been on that.
Thank you.
you are so welcome. I
Lord.
think that is.
That's gonna be awkward.
It's gonna be so awkward.
Thanks.
But I guess.
Where'd the other day?
So oh wait, let me see.
So my friend told me. That's my dog. It's right here.
Yeah, is that your blood?
Avery.
Mm-hmm.
Sucks.
Funny.
I want to see a picture.
I just haven't worn my hair up or down. So
I haven't either.
I always wear it down and then the same as it up. I'm gonna bring mine too. We're showing off Sweetheart's pictures, so. There's that.
Holding hands. Touching me. Touching you. Sweet. You know?
Bum, bum, bum.
You said. Do you want to see the cutest video ever? I didn't think it was the cutest video ever.
undo them, get them spread out so you can see. Hopefully they're growing. Yep, everybody needs a flashlight over here. Everybody got a UV flashlight right here? Ah, something's wrong. Everybody get a UV flashlight. Okay, I'm gonna turn off the lights. Okay, try. You can give it a go.